Welcome. Today we're exploring an innovative strategy to combat neurodegeneration, the targeted blocking of the cGAS-STING signaling pathway. But first, what exactly are signaling pathways in our body? Signaling pathways are essentially the body's internal communication network. They consist of a series of chemical reactions that allow cells to respond to environmental cues, such as hormones or neurotransmitters. These pathways regulate a wide range of functions, from our heart rate to our mood, playing a crucial role in our overall health. The cGAS-STING pathway is a key component of our body's immune response. When our cells detect misplaced DNA, a potential sign of infection or cellular damage, the cGAS enzyme springs into action. The cGAS enzyme produces a molecule called cGAMP, which then activates the STING protein, initiating an immune response and the release of interferons, which are essential proteins for defending against viruses. This signaling pathway is not only vital for our immune response, but it also plays a significant role in neurodegeneration. It identifies DNA in the cell plasma, also known as the cytosol, and triggers inflammation and the release of interferons. These proteins are essential for defending against viruses. In neurodegenerative diseases such as Alzheimer's and Parkinson's, this pathway can become activated, potentially due to stress in the mitochondria, the powerhouses of our cells, and leakage of DNA. This activation leads to neuroinflammation, or inflammation within the nervous system, through the production of type 1 interferons, contributing to neuronal damage. While the cGAS-STING pathway is essential for fighting off infections, it can also contribute to neurodegeneration when overstimulated. In conditions like Alzheimer's disease, persistent activation of the cGAS-STING pathway can lead to chronic inflammation and neuronal death. This complex interplay between our immune response and neurodegeneration underscores the complexity of these diseases and the importance of ongoing research in this field. Now, how can the targeted blockade of the cGAS-STING signaling pathway help halt neurodegenerative processes, particularly in old age? In neurodegenerative diseases, activation of this signaling pathway triggers neuroinflammation through the production of type 1 interferons, leading to neuronal damage. By inhibiting STING, a crucial protein in this pathway, with compounds such as C176, it's been demonstrated that neuroinflammation can be suppressed, dopaminergic neurons can be protected, and motor deficits can be improved, as seen in models of Parkinson's disease. Moreover, activation of the pathway due to mitochondrial stress and DNA leakage contributes to downstream neuroinflammation, positioning it as a potential therapeutic target for various neurodegenerative diseases. This highlights the promising potential of targeted interventions in the cGAS-STING pathway in the fight against neurodegeneration. Interestingly, natural compounds and lifestyle factors can also influence the cGAS-STING pathway. For instance, licorice extract has been found to inhibit pathway activation by targeting STING oligomerization. Additionally, impairment of Golgi transport can activate tonic interferon signaling, leading to an increased immune response and protection against infection. Conversely, Golgi vesiculation during overt mitosis can limit cGAS-STING responses, suggesting a secondary means of preventing hyperactivation. This highlights the potential of natural compounds and cellular mechanisms in regulating the cGAS-STING signaling pathway. Research suggests that obesity and related metabolic disorders may activate the cGAS-STING pathway, leading to an increased immune response and potentially contributing to the progression of neurodegenerative diseases like Alzheimer's disease. Furthermore, the cGAS-STING pathway is crucial for innate immunity. It recognizes both foreign DNA and host DNA to trigger antiviral responses. In addition, the STIM1 protein, which is involved in calcium signaling, can activate STING through altered intracellular Ca2+ levels, thus linking it to the cGAS-STING signaling pathway and the progression of conditions like atherosclerosis. Overall, lifestyle factors such as exercise and diet can influence the cGAS-STING pathway through their effects on metabolism, inflammation, and cellular responses. This highlights the complex connection between lifestyle habits and immune signaling pathways. In conclusion, targeted blocking of the cGAS-STING pathway holds promise for developing treatments for neurodegenerative diseases by modulating inflammation in the central nervous system. We hope you found this information enlightening and look forward to sharing more exciting developments in this field. Thanks for joining us, and as always, we wish everybody a long, healthy, and happy life. See you next time.